Our next awardee is our 2025 Hannan medalist. This is a career award that recognises outstanding research in any of the fields of statistical science, pure mathematics, applied mathematics, and computational mathematics. It honours the contribution of time series analysis of the late Professor Edward Hannan, Professor of Statistics at the Research School of Social Sciences at the ANU. And this year, we congratulate Academy Fellow Professor Noel Cressie. Professor Noel Cressie is a world leader in statistical science for the analysis of spatial and space-time data, especially in environmental science. He has made pioneering, fundamental, paradigm-shifting and highly influential contributions to theory, methodology and applications. Professor Cressie is a leading exponent of statistical methods in environmental science, especially for large-scale phenomena such as oceanic and atmospheric circulation and climate. By combining physical principles with stochastic models to capture uncertainties, he has developed a powerful methodology to investigate causal links in these complex global processes. He has overcome huge challenges to make the methodology computational feasible for large complex data sets. Professor Cressie has played a key role in applications to global CO2 flux, regional climate, sea surface temperature, air pollution, disease mapping, biogeochemical cycles, soil carbon dynamics, movement of glaciers, and river pollution. So you can see he has very broad interests. His recent work on climate model uncertainty may ultimately have a substantial impact on science and policy. Congratulations, Professor Noel Cressie.
Thank you.
Congratulations.
Well, thank you to the Academy. What an amazing honour to be named the Hannan medalist. I'm really privileged. I look upon it as my first Academy Award, and I want to call it the TED. So I'm very pleased to get a TED. I would like to go through my journey just a little bit. I started out at the University of Western Australia, and Don McNeill actually was there for a short time and suggested I apply to Princeton. On my way to Princeton, I stopped at the ANU for 6 months, and there I got to know Ted. I was at Princeton for a while. I was a Fulbright scholar, actually, and there I had advisors of Jeff Watson, John Tukey, and had some postdocs. Came back to Australia for a few years to Flinders University, and then was offered jobs in the Midwest of the United States, and was probably thinking of staying there for about 5 years, and 30 years later came back to Wollongong with the urging of David Steele, and have been director of the Centre for Environmental Informatics since then. All right, so as Monty Python says, now for something completely different. I thought we'd go to a different planet, that we go to Mars, and this represents some work that I've done in the last year and a half with NASA and the European Space Agency, ESA. My story starts 4.5 billion years ago when the solar system was very young, and there was a lot of exoplanets and asteroids smashing into Mars and Earth for that matter. And with a small stop about 1 billion years later in the Jezero crater, we go to the present. So NASA launched a mission to Mars, and it was the Perseverance rover, and it landed on Mars on the 18th of February 2021. The Jezero crater was active about that billion years later, about 3.6 billion years ago, and there was water and river valleys. And the thought was that the rover would land in the Jezero crater where there was a possibility of life because of all that water. And there is still water underneath the surface of Mars. The goal, one of the major goals, was to search for ancient or possibly extant life. And currently the rover is drilling into the rock. It's also collecting rubble. And in these tubes, these sample tubes, which are being stored in the rover, it's also collecting atmosphere. So there's an excellent sort of choice, if you like, of where that life might exist or might have existed. This is a very ambitious project. It's called Mars Sample Return. And the idea is to send a rocket from Earth to Mars, and in the rocket there would be another rocket which would collect the sample tubes, take off from Earth and end up sending the samples back in a package that would land in the Nevada desert in about 15 years. As you can imagine, it's very ambitious and actually quite expensive. But we have that mission, and I was honoured to be asked to be part of the Sample Safety Assessment Protocol. This is a planetary protection group that is an attempt to keep the Earth safe, or Earth safe, from possible potential biohazards. And my presence on there was as a discussion leader in statistical science, and there I am in the group. One of the key deliverables of this group was to put together a protocol to keep Earth safe, and we developed what was called a Tiger Team. Now, NASA develops Tiger Teams to get onto problems really quickly and to get out of them again. So the Tiger Team disbanded late last year, and we're very pleased to learn that we actually won a NASA award, the Group Award for Research at NASA, in June of this year. It was really a wonderful experience to be part of a very interdisciplinary team: astrobiologists, geologists, virologists, and one statistical scientist. And in there we have about, in each of the tubes, and there's 30-odd tubes, 10 to 27 grams of material. Now, those grams of material actually have to be used for something, for science. However, it also has to be used to keep the planet safe, and you can see there's a tension between testing, often destructive testing, to keep the planet safe, and leaving enough of that material behind for science. And that's where I got involved. So this whole idea that we developed is centred around a three-step protocol. First of all, you get heterogeneity, you work out where you might sample in the tubes to take the subsamples. Then there's the notion of an abiotic baseline, and if you exceed the abiotic baseline, then by the way, all of this is in a sample receiving facility, which is built specifically in the Nevada desert to do this sort of planetary protection work. If the molecules look biologically oriented, then you would then do further testing to see if they're hazardous or not. The challenge would be to determine the number of subsamples needed to test this hypothesis, the absence hypothesis, no biohazards, versus the presence hypothesis, biohazards. And the optimization that I was involved with was to use as little of the returned material as possible, but not so little that we would not keep the planet safe. So you can see there's some sort of existential questions going on here. And so what I developed was a criterion which I call the Bayes error, or BE, which is a function of the number of subsamples in each of the tubes. So we did this tube by tube. But we have to know what it means to keep the planet safe. And so NASA, ESA, specify a risk tolerance threshold, which I've called P star, and typically that's 1 in a million. But I could have developed it and I have developed it for smaller and higher thresholds. First of all, as a statistician, and I'm sure you know that nothing is certain, and Oliver Cromwell tried to relay that to the Scots before he defeated them at the Battle of Dunbar, and also the famous Benjamin Franklin quote that there's nothing certain except death and taxes. So what happens is we do that heterogeneity. We look at the types of rocks that are in the samples. We look at the atmosphere, the regolith and various rubble and things. We have these two models, the absence model and the presence model, and we worry about false positive errors and false negative errors because nothing is as certain. So the false negative error is the one I think that should get more attention, and I certainly concentrated on that in the work that I did. It says that we've found no biohazard, but in fact there is a biohazard present. The false positive error would be, if you like, less consequential, that you've declared a biohazard to be present, but it's not present, and perhaps further testing would resolve that. But if, in fact, you've declared no biohazard and there is a biohazard present, perhaps you've then released these biohazards onto our planet. And as I mentioned, there's this tension between using enough so that we keep everything below the safety sample threshold, but leaving enough for science at the same time. So as a Bayesian, I thought we need to introduce whether these models are more realistic or not, the absence model versus the presence model. And so I used pi naught to represent the probability that there is no biohazard present, and pi 1 the probability that there is a biohazard present, and pi 1 is equal to 1 minus pi naught. We also have measurements. These are measurements that haven't been taken yet, and this is partly the problem of this. This is a design problem. We're pre-measurement. We have to design, it sounds impossible, but it's possible. We have to design something where we have no measurements. And so the distribution of these measurements, which we know are uncertain as well, there's uncertainty in those, are called likelihoods. And so under each model, M0, we have a likelihood F0, and under the model M1, the presence model, we have a likelihood F1. These are the components, if you like, as I'm building up this notion that there is uncertainty everywhere, and I'm trying to then characterise it as a statistical scientist. I've mentioned that this is a design problem, and what I developed was a Bayesian design criterion, which I called the Bayes error. And it is, in a sense, the expected error, the false positive error and the false negative error weighted by the priors pi naught and pi 1. This is the sort of thing that you get as a cartoon, obviously, but it's showing the Bayes error on the vertical axis. It shows P star, the risk tolerance threshold that NASA tells us, 1 in a million, for example. And then as I take more and more samples, that error decreases, as you would expect. So the Bayes error keeps going down. I can keep driving it down as far as I can, more and more samples until I run out of samples. But clearly what I want to do is drive it until it's just below that threshold, that I can then say 1 in a million, we've achieved that. And I use that number, in this case, it's n star equal to 5. So what happens is that this optimal n star actually depends on a number of key quantities. And it just takes building the model to work out what those key quantities are. Pi naught I've already mentioned, it's the prior. Something called kappa, which I've only indirectly referred to, and that is the sort of loss you would make if you had a false negative error versus the loss you would make if you had a false positive error. And the ratio of those two, and we keep kappa small in this case, is really important, and it's an important dial, if you like, which determines how large n should be. The last one is the distance, so-called distance or divergence, between those two likelihoods under the two different models. And how would you get a hold of this divergence? Well, there are actually experiments around that are attempting to come up with Mars-like materials that would be biotic and abiotic, and there is such a paper which we use to then build the model in a principal component space. This is what it looks like after you do the sort of fitting that statisticians do, and that's a normal distribution in 2 dimensions, the principal component dimensions, with both the abiotic and biotic on the same graph. So the important part of this particular slide is the second dot point, which is how do you calculate this Bayes error? Well, it's possible to do it analytically, but remember these 3 stages of the protocol that we've developed. Each one leads to the other. And so there's actually a sequence of tests of models. And it became clear to me that the way to do this was to pretend to be in the sample receiving facility and construct a digital twin of what was going on, and then using all of this information to come up with the appropriate dials, the kappas, the deltas, and the pi naughts. Okay, the chairman, who managed to herd a whole bunch of cats, including a statistical scientist, called this the money plot. And it's very American, you know, to think about the money plot. That's where the money is. That's what's going to get NASA really excited, and it did. This is the money plot where I get to plot pi naught and delta, and I hold kappa fixed, because I've only got 2 dimensions on a money plot. And I have my P star, which are the red dotted lines, 1 in a million. I then work out under these different circumstances, different pi's and different deltas, what n star would be. And it can be 1 if your deltas are very far apart. And as the deltas get closer together, the number that you need gets bigger. And I gave him some numbers which had, I don't know, 15 or 20, and they were a bit unrealistic. But on this particular money plot, the largest n star that I came up with per sample tube was 5. And then one works out how much of that sample that you would need. This is a plot to just show you what happens as you artificially move those 2 likelihoods closer together. And as you would imagine, as they get harder to determine which model is correct, the number that you need goes up. So n star keeps going up and up. But basically, we're sitting at around 5. So this is my Hitchhiker's Guide to Mars. And of course, it's not to the galaxy. It's only a smaller place. It isn't 42, it's 5, but with a plus or minus.